Assalamualaikum warahmatullahi wabarakatuh and very good day. Today we are from group 3, want to present about control of dirofilariasis. So what is dirofilariasis? Dirofilariasis is a disease caused by Dirofilaria, which is the parasitic roundworm that affect variety of mammals such as human, dogs, even cats. It also known as heartworm diseases. It's actually infect through the mosquito's bite. So it is important for us to take precaution against the mosquitoes to reduce the transmission of lymphatic filariasis and other mosquito-borne infection. So here we are discussing, we are dividing into two, which is biological control and non-biological control. Biological control is usually involve natural enemies and involve active human role. Biological control have benefit for the environments and other non-target organism. For non-biological control, is involve non-sustainable options and it use chemical but detrimental for living organism. So here we are discussing about 5 main points, which is 3 from biological control and 2 from non-biological control. So for biological control, the first one is mosquitoes predator. So the first predator is larvivorous fish. Is effective at reducing mosquito larva population in water, as the fish is actually eat the larva stages of mosquitoes. Larvivorous fish also live in varieties of habitats. So the second predator is larva of elephant mosquito. Did you know that there are mosquitoes that eat on another? So the species is called as Toxorhynchitis species. It also known as elephant mosquitoes because of its size. It's larger than other average mosquitoes. As the adult form of mosquitoes, it doesn't consume blood and it harmless to human. So next I will pass to the next presenter.
Okay, now I'm going to talk about the plant-borne mosquitocytes and repellents. Plant-borne mosquitocytes are often effective at a few parts per million against AIDS, Anopheles, and Culex, young larvae instars. 18 plant species have been employed for the successful synthesis of nanomosquitocyte. For example, Loxferol is a contains polyphenols, phytosteroids, indoles, etc. Its bitter juice is used for repelling flies and mosquitoes. The second example will be Thymus vulgaris. Thymus vulgaris is an herbaceous plant which contains essential oil that contains thymoid, carbachol, linalool, and chiaphalin, which have insecticidal properties. Laboratory studies showed efficacy of the essential oil against mosquito larvae. Thyme seems to have anti-helminthic properties as well. Next.
Okay, so I'm going to talk about genetically modified mosquitoes. So female mosquitoes will usually ingest the first stage larva and it will develop inside its body, and then the L3 larva will be inoculated into a susceptible host when the mosquito feeds. So in order to control the spread of Dirofilaria, we can modify the mosquitoes genetically. So either to prevent the agent from maturing inside the mosquito, or from preventing the mosquitoes to reproduce properly. So researchers have created modified genes that they insert into male mosquitoes so that those genes will create proteins that will inhibit the growth and the development of like mosquitoes. So when the male will reproduce, all its offspring will have the same protein, which will prevent them from becoming adults and will kill them off, which will reduce the population of mosquitoes and thus reducing the spread of Dirofilaria. Next.
So I'm going to present about the non-biological control of Dirofilariasis. Next. So for the first point is regular use of macrocyclic lactones. So there are many more actually, but um, these are some of it. So the first one is selamectin. Selamectin is a monthly application of selamectin in spot formulation have been used successfully to reduce the transmission of pathogens under natural field conditions. When infected animals are treated twice a month, the dog's protection period is extended to 9 months. Other than that, there's a milbemycin oxime. Milbemycin oxime, which is given orally once a month, may be effective in protecting dogs from subcutaneous dirofilariasis in endemic areas and may provide a more chemical resistant options. Last but not least, use moxidectin. So the use of moxidectin in sustained release formulation administered subcutaneously was found to have absolute efficacy in the prevention of Dirofilaria repens in an experimental trial. And the author suggested that excellent effect of the formulation is largely due to lipophilicity of ingredients stored in the body fat. Next. So these are the macrocyclic lactones. So you can see these are the active ingredients for dogs. And these are the formulation, chewable, injectable, tablet, and spot on. And also there are the dosages for dogs. Next.
Last but not least, mosquito larval control method can help to control the Dirofilaria. Mosquito is the intermediate host that will transmit Dirofilaria from one host to another host. Thus, this method helps to control the population of the mosquito. As we know, mosquitoes usually breed in stagnant and standing water. Therefore, this method aims to eliminate water breeding sites that cannot be drained and filled, consequently will prevent the increase of its population. Mosquito larvae can be controlled through the use of low-impact larvicides that selectively kill only mosquito larvae. Nowadays, in the market, we can find larvicide in several forms such as powder, tablet, liquid, and include methoprene monomolecular superspeciculum, larvicide oil, chemical insecticide, neurotoxic insecticide, plant-derived product, and larvicide bacteria. Next. Here is the two examples of larvicide commercially sold in the market in form of liquid and also powder. Next. With that, our presentation ends. Thank you.